Greetings biochemistry students, and welcome to this PowerPoint lecture about electron transport and oxidative phosphorylation. This is Dr. Michael Garut. This is a summary of the electron transport chain. In the picture, you're looking at a simplified view of a mitochondrion with the matrix being this sort of melon color in the center, the intermembrane space being out here in the white part, and then the inner mitochondrial membrane being this yellow and blue lipid bilayer. The inner mitochondrial membrane is packed full of these enzyme complexes, 1, 2, 3, 4, and 5, which we'll see what they do specifically. These enzyme complexes 1 through 4 are involved in taking the energy from our reduced cofactors in terms of their electrons in redox reactions, transferring those electrons eventually to oxygen, which is the final electron acceptor, and then harnessing that energy to pump or transport protons or hydrogen ions from the matrix out into the intermembrane space. Then that is the source of energy that can be used. We can call this the chemiosmotic potential, the electrochemical potential, right? We have a higher concentration of protons on the outside, which means a concentration gradient and also a charge gradient, hence electrochemical. That source of energy, as those protons are energetically favored to go back into the matrix, that energy is harnessed to do a phosphorylation of an ADP with an inorganic phosphate. Therefore, this is called oxidative phosphorylation because of the oxidative reactions that are coupled with the phosphorylation from this proton gradient. The oxidation and the phosphorylation are coupled. That's why we talk about the electron transport pathway and oxidative phosphorylation always together. They are separate processes, but they're coupled. If one doesn't work, the other one also will not function. This is a diagram that is very similar to this diagram, but only showing a cross-section of the membrane and a little bit more detail about where the electrons are transferred and where the protons are pumped. The number of oxygens that are used up from one NADH, according to this diagram, is half of an oxygen. That means it would take 2 NADHs donating 4 electrons in order to use up a full oxygen, 4 protons, and make water. That's the stoichiometry. Then, how many ATPs can be produced for one oxygen molecule that is reduced, or specifically one oxygen atom that's reduced? That is referred to as the P to O, or just the PO ratio, or sometimes the P to 2 electron ratio, but that's much less common. Going back to the stoichiometry, 2 electrons come from an NADH, then that allows you to reduce just 1 oxygen atom to water. So that's 2 electrons. How many phosphates can you attach? How many high energy phosphates can be created is the PO ratio. That doesn't have to be a whole number because it depends on how many protons are pumped out of the matrix and how many protons it requires to generate an ATP. You can calculate the delta G of that. We won't be doing that in this class, but it can be done. In the electron transport chain, you're doing a series of redox reactions, and each one of those reactions needs to be energetically favored, have a negative delta G or a positive delta E. That is a more positive reduction potential. And in order to make that happen, you have many different electron carriers along the pathway, and those electron carriers make use of many different coenzymes or cofactors. Many of these come from vitamin sources, so we'll see where those are used in the various steps. Flavoproteins, you can imagine that comes from a flavin, which is riboflavin. Here's a diagram I found online and modified just a little bit discussing the various B vitamins. Flavins, or riboflavin, are vitamin B2. That gives you a precursor for FAD, which stands for flavin adenine dinucleotide. Thiamine is vitamin B1, which gives you the thiamine pyrophosphate. Niacin, vitamin B3, which is a precursor for nicotinamide adenine dinucleotide, or NAD. B4, which isn't normally called a vitamin any longer, is adenine, so flavin adenine dinucleotide, nicotinamide adenine dinucleotide. Coenzyme A also contains adenine, and it contains pantothenic acid, so coenzyme A uses both of these. Pyridoxine is a precursor of pyridoxal phosphate, which we'll see later in the course. B7, I didn't know anything about this. It's a sugar. B8 is biotin. So since B7 apparently is no longer considered a vitamin, sometimes biotin is called B7, but really we just call it biotin. B9 we call folic acid, right? We don't call it B9. But B12 we definitely still do. That's cobalamin, and we'll see where that comes in also later in the course. Coenzyme Q, which is also called ubiquinone, you can tell it has a long hydrocarbon tail, making it anchored into a lipid bilayer, and then it has this quinone portion. A quinone is a diphenol in a reduced state that has a diketone that is conjugated in the oxidized state, and there's the semiquinone intermediate, so these can facilitate one electron transfers, whereas NADH typically only does 2 electron transfers. Cytochromes are heme-containing proteins. They can also facilitate 1 electron transfers, in which iron oscillates between an iron 3 and an iron 2 state. Iron sulfoproteins, same thing. Also, iron oscillates between positive 3, positive 2. And if we start with the NAD reaction, notice that we are going from a negative towards a positive, or each time going downwards towards a more positive reduction potential. So each of the reactions that you see, skipping this one for now, but it's about the same. NAD transfers enzymes to FMN, to coenzyme Q, to cytochrome B, C1, C, A3, and finally to oxygen. This is not listing every single step of the electron carriers, but some of the major ones. There is something called the Q cycle, which is how the coenzyme Q goes back and forth between the reduced and oxidized forms. In complex 1, the NADH donates its electrons right here to this flavin mononucleotide, and each of these orange and red images are the different iron sulfur centers that the electrons are transferred through in increasing reduction potentials. 2 protons from the matrix bind or bond to coenzyme Q to make coenzyme QH2, so that reduces the number of protons in the matrix, contributing to the chemiosmotic gradient. So there's a complicated stoichiometry there, because NADH uses up a proton. 4 protons are pumped out, so the stoichiometry is somewhat complex, and for our purposes, if we just consider 4 protons being pumped, that's sufficient for our understanding. Complex 2 does not pump any protons, and complex 2 is also the succinate dehydrogenase complex that doubles as a citric acid cycle enzyme. Complex 2 also reduces coenzyme Q to coenzyme QH2, so from this point, the pathways from NAD and FAD converge. In complex 3, coenzyme Q transfers electrons through various cytochromes, those are heme-containing proteins, eventually to cytochrome C, which is a soluble electron carrier. And in complex 3, which we're looking at here, 2 protons are pumped in this complicated Q cycle that you're not responsible for. In complex 4, then, the final electron transport complex, cytochrome C brings the electrons over and transfers them through some various cytochromes. You can see the hemes here. These would be copper ions in the various subunits that are also part of the electron transport chain. And in that pathway, 4 more protons are pumped from the matrix to the intermembrane space. Again, you're not responsible for the details of the electron transport mechanism. One interesting discovery is that many of these enzymes complex together in the membrane of the mitochondria into multi-enzyme complexes referred to as respirasomes, which contain all of the proton pumping enzymes 1, 3, and 4, and these are large enough to be seen with electron microscopy. Here are 2 diagrams that sum up the flow of the protons and electrons. One from an older version of your text that shows more of the secondary structures of the 4 complexes with their alpha helices, and one newer one that shows less of the secondary structures, but more detail on the pathway of the electrons, the Q cycle, and the protons being pumped. So while it is complicated to accurately quantify the numbers of protons that are pumped and the numbers that are consumed, which is different than pumping, they're showing in this picture hydrogen ions being consumed from the matrix. It's fine to just think about it as 4 protons being pumped by one complex, 4 by another, and 2 by another. At any stage of the electron transport pathway, if you have a molecule that inhibits that particular stage, then it's going to block the electron transfer at that point. Some of these are pharmaceuticals that are drugs that may be used in humans, may be used to kill insects, or antibiotics. Since the proton gradient that is created from the electron transport chain is obligately coupled to the oxidative phosphorylation, anything that can diffuse that proton gradient without making ATP is called an uncoupler. Dinitrophenol is the earliest and most famous example of something that was studied as an uncoupler. Because of its structure, it is nonpolar enough. The electrons, even in the conjugate base, are spread out by resonance to all of these other oxygens. So they're so diffuse that this is in fact able to traverse a lipid bilayer. If it can do that, then it can pick up a proton on one side and it can liberate that proton on the other side. When that happens, the protons that have been pumped out can be carried by that uncoupler back into the matrix. That disseminates that proton gradient without producing ATP. That can make it a diet drug. It also is going to, since the energy is not being saved as ATP, is going to generate some heat. The difference between an effective, quote, diet dosage and the dosage which would kill a cell because it can't generate ATP is just way too close together to consider actually using these as pharmaceuticals. The rest of the PowerPoint discusses complex 5, also known as ATP synthase, which is the enzyme that actually phosphorylates ADP to make ATP. This enzyme is known to contain 2 distinct components, FO, not F0, even though many videos and papers often refer to it as F0. The O is for the donut-like form that has a pore for protons, and then an F1 unit. Before we look at this top view, let's look at some of the overall structure of the ATP synthase enzyme. Okay, here's an overall structure of that ATP synthase enzyme. Let's look at this one. So the N side and the P side. I haven't referred to those earlier. Notice that the N side is the matrix and the P side is the intermembrane space. So the electron transport chain is pumping the protons from the N side to the P side, and here is the ATP synthase complex. There are many subunits, alpha, beta, gamma, etc. we'll look at in a minute, but the part that is embedded within the membrane, this circular part, that's the FO complex. It's connected by a central shaft to the F1 portion of the complex, which is up here. We also have this linker arm, which stabilizes the complex in the membrane so that when this FO complex rotates around, linked by this gamma shaft, that the F1 part does not rotate. But this gamma shaft, where you can see the little tiny bit of that color right here, rotates inside the F1 complex. So that's an overview. Here it is with the secondary structures showing this linker arm with a bunch of alpha helices. Here's your FO complex, and that FO complex has these proton binding sites right in the center on each of the alpha helices. So the F1 component has 3 alpha subunits, 3 betas, a gamma, a delta, and an epsilon. Each of the beta subunits has an active site where you can synthesize an ATP. So what we're looking at here in this top view are the alpha and beta sites. Alpha, beta, alpha, beta, alpha, beta. And each has a different conformation. As the linker arm rotates, that is what's in green, as that rotates, these purple and gray subunits, the alphas and betas, they don't rotate, but they do change conformation. So let's start down here at the lower left in the empty subunit. This is also sometimes called the loose or the open. The empty or open active sites have no ADP or ATP bound. Then when this internal protein rotates, the conformations of these change to be like these. In that case, we have the loose site. This is the one that allows the ADP and the phosphate to diffuse into the active site. Here's the ADP that has come into the active site. Then when this central shaft turns again, the conformation changes to squeeze together the ADP and the phosphate. That's the tight binding site, and that forces the phosphate and the ADP to attach. Then when that site becomes the loose site again, then the ATP that has been formed can dissociate. Now looking at this diagram and the way I've described it, you might think that this is rotating around, but it's not. And a different diagram is perhaps an easier way to see that, which is this diagram, which shows that the subunits stay in the same place, but they just change conformation. So if we start, let's say right here, if this is the open where the ATP can diffuse out and become empty, then this one, going around clockwise, then this one at the bottom can become this one, which is now the loose binding where it can bind the ADP and the phosphate. Then it can change the conformation to the tight binding where it forces the ADP and the phosphate to bond together to make ATP. And then when it changes conformation one more time into this state, it becomes empty again, or the open state. If you look at each of the subunits, they require, well, according to this diagram, roughly 3 protons to change conformation each time. So 3 protons, 3 protons, 3 protons, 9 protons, but 3 ATPs are produced. Now let's talk about the F0 complex. So the F0 complex is this ring. It's also called the C ring because it's made of the C subunits. You have the alpha, beta, there's delta, there's gamma, there's epsilon, and then A and B, but then here you have the C ring. So for the FO subunit, you have the A subunit, the B subunit, and then the C ring. The C ring can have from 8 to 17, or sometimes even more or fewer than that, as we'll see, subunits. So the one we're looking at here, 1. This diagram shows 10 subunits. So that means there are 10 proton binding sites, one in each of those C subunits in the ring. Here's your B linker arm that keeps the F1 subunit from rotating. So in this model, the protons are diffusing in from the P side, binding to one of these subunits, and when they bind to it, it is then allowed to rotate away from the A protein, in this case, in a clockwise direction. This was famously discovered, and the evidence for that was done with an experiment in which an actin filament and a fluorophore were attached to this C ring, and then the enzyme was run in reverse by putting ATP into the system, which causes the C ring to go in the other direction and ATP to be hydrolyzed. And then you could look at this in an electron microscope and literally see this fluorescent actin filament spinning around in real time. It's not known exactly how the flow of protons causes this ring to rotate, but this is the best going model. You've got in each of the C ring subunits, between them is an ion pair with a glutamate and an arginine. When the proton comes in and protonates that glutamate, I said glutamate residue to become glutamic acid, that can trigger a conformational change, and that conformational change can trigger the rotation. The result of all this is that the number of protons that are required depends on the number of subunits in the C ring. And since the number of subunits in the C ring varies from species to species, that means the number of ATP that can be generated per NADH or per oxygen atom is different. Therefore, the P to O ratio, let's call that X in this, in humans is about 2.5 for NADH, 1.5 for FADH2, but it varies depending on the number of C units in the complex. Here are a couple of examples of fluorescent confocal microscopy showing differing numbers of C units in the complex. These two have 13 or 14, and I looked up some research articles about the total numbers of those. Notice that humans have 10, as do yeast. So yeast are a good model for human ATP synthase. Bos taurus, that's cows, they have 8. Very interesting. So that means they would get even more ATP per proton. Many have more subunits in their C ring, so the number of subunits differs by species. And then there are a few bacteria that instead of using protons, in fact, can use sodium ions to generate ATP. So the last thing to take a look at is the difference in how NADH that is created in the cytosol can get into the mitochondria to be a part of the electron transport chain. NADH itself does not cross the mitochondrial membrane, so therefore we need to have some kind of a shuttle system to get the reducing equivalents across the membrane. There are a couple of different ones that we'll take a look at: the malate aspartate shuttle and the glycerol 3-phosphate shuttle. The glycerol 3-phosphate shuttle, as you can see here, operates in skeletal muscle and the brain. Here's a diagram. NADH that comes from glycolysis transfers its electrons to dihydroxyacetone phosphate, which is also a glycolytic intermediate, to make a glycerol 3-phosphate. And glycerol 3-phosphate can be used in this mitochondrial glycerol 3-phosphate dehydrogenase to reduce ubiquinone to coenzyme QH2. In this case, an NADH in the cytosol is equivalent to an FADH2 in the matrix, meaning you only get 1.5 ATP per NADH that enters via this shuttle. However, it does only require one additional enzyme, this glycerol 3-phosphate dehydrogenase, one in the cytosol and one associated with the matrix. The malate aspartate shuttle is more efficient. It requires a few more enzymes and a transporter, but it does give you the full amount of ATP. This takes place in the heart and liver and kidney, which are tissues in which you want to get all of the energy possible from the NADH. This shuttle requires 2 transport proteins, one which is a glutamate aspartate transporter. Take a look at the structure of aspartate and glutamate. You'll notice that they're very similar but just differ by 1 CH2. And then you have a malate alpha-ketoglutarate transporter. Take a look at those and you'll see the same thing. They're very similar in structure. They differ only by 1 CH2 and 1 carbonyl versus alcohol. Those transporters, those are additional proteins that are required. In the malate aspartate shuttle, you also use some of the citric acid cycle enzymes and some enzymes of amino acid metabolism to take the electrons from NADH to oxaloacetate to make malate. The malate is transported and then the NADH is regenerated. If that were all that occurred, it would be a net transport of NADH, but also of malate, and that would cause malate to build up. So then the rest of this is a way to recycle the malate back to the outside by going back to oxaloacetate and then exchanging with glutamate and alpha-ketoglutarate to aspartate back to the matrix. Back to the intermembrane space. You're not responsible for knowing the details of this shuttle, just that the type of shuttle that is used affects the number of ATP that can be generated per cytosolic NADH. That makes it 3 if the glycerol phosphate shuttle is used, or 5 if the malate aspartate shuttle is used, therefore giving you a slightly different number of ATP depending on the tissue and which shuttle is used. Here's an accounting of all of the ATP generating reactions in muscle using the glycerol phosphate shuttle. And then just 2 slides at the end. What happens if some of these electron carrying complexes leak out of these complexes and reduce oxygen to superoxide? So this is O2, which has a negative charge and an extra electron. So that's a radical anion. Oxygen readily gains an electron like this, and when that happens, it needs to be detoxified immediately because it's extremely reactive. There's an enzyme that's present specifically to degrade these reactive oxygen species. That enzyme is called superoxide dismutase. This is superoxide. And a dismutase is an enzyme that causes a disproportionation. That is, you take 2 semi-oxidized thing and you turn it into 1 reduced thing and 1 fully oxidized thing. So it transfers those. And what's less toxic than superoxide is hydrogen peroxide. Hydrogen peroxide itself is still toxic, and you have another enzyme that's called catalase that degrades the hydrogen peroxide back to water and oxygen. So you end up with those 2 enzymes taking the superoxide and turning it back into oxygen. This enzyme is also present pretty much everywhere, and it is the reason that when you have a cut and you put hydrogen peroxide on the cut, that you see bubbles forming. You have catalase in your blood, and the catalase takes the hydrogen peroxide, converts it into oxygen gas and water, and the oxygen gas are the bubbles that you see. So this diagram, which isn't in your textbook, but I took from reference, Redox Biology, Volume 37 from October 2020, these red numbers 1 through 7 are showing the most common sites that the superoxide is generated, and there are 2 different superoxide dismutases, 1 in the intramembrane space and 2 in the matrix. There's the catalase that I mentioned, and many other proteins that are involved specifically in the degradation of these reductive oxygen species. This diagram, which is in your textbook, indicates that one of the common molecules involved in these steps is glutathione. So glutathione is an enzyme that has a thiol group that can be oxidized to a disulfide group, and those then can therefore be used to get rid of the reductive reactive oxygen species. That's the end of electron transport, oxidative phosphorylation, and ATP synthase. Cheers.